Welcome to this lecture on the biochemistry of metabolic processes. Today, we are going to explore one of the most elegant and crucial mechanisms in all of biology, the chemiosmotic hypothesis. If you have ever wondered exactly how the food you eat is converted into the energy that powers your heartbeat, your muscles, and your brain, you are in the right place. Let us dive in and build our understanding from the ground up. Before we look at the complex biology, let us start with a simple analogy. Imagine a massive hydroelectric dam holding back millions of gallons of water. That water desperately wants to flow downward due to gravity, creating immense potential energy. When we open a small gate, the rushing water spins a mechanical turbine to generate electricity. The chemiosmotic hypothesis describes a nearly identical process happening inside your cells every single second. But instead of water, your cells use tiny charged particles called protons. Instead of a concrete dam, they use a biological membrane. And instead of a metal turbine, they use a spectacular spinning protein called ATP synthase to generate ATP, the chemical energy currency of your body. Now that we have a mental picture of a dam, let us look at the formal scientific definition. The chemiosmotic hypothesis was originally proposed by the brilliant scientist Peter Mitchell in 1961. It is the scientific model that explains exactly how energy derived from electron transfer is ultimately used to manufacture ATP. Formally, the hypothesis states that the transmembrane flow of protons, moving down their concentration and electrical gradients, provides the free energy necessary for the synthesis of ATP. In simpler terms, the cell creates a gradient of protons, and letting those protons flow back across the membrane releases the energy needed to keep you alive. So how does the cell actually build this biological dam? During cellular respiration, specialized protein complexes sit within the inner mitochondrial membrane. These complexes act like powerful water pumps. As electrons are transferred through the system, these complexes use that energy to actively pump hydrogen ions, which are protons, across the membrane. Specifically, they force these protons from the mitochondrial matrix up into the intermembrane space. Because the membrane is strictly impermeable, the protons cannot simply leak back across. They are trapped there, building up a massive biological pressure. This continuous pumping of protons does not just create a simple physical buildup. It creates two distinct gradients at the exact same time. First, because protons are hydrogen ions that determine acidity, their buildup creates a chemical gradient, meaning there is a significant difference in pH between the two sides of the membrane. Second, because every proton carries a positive charge, their accumulation creates an electrical gradient, or a voltage difference. Together, this combined electrochemical gradient creates a powerful driving force that desperately pushes the protons back toward the matrix. You might be wondering, why is this specific mechanism so incredibly important? To truly appreciate this concept, we must understand what would happen without it. If your body only relied on the basic breakdown of glucose, a process called glycolysis, you would only produce 2 molecules of ATP per glucose molecule. That is barely enough to keep a single-celled organism alive. However, by utilizing the chemiosmotic mechanism in our mitochondria, that same single molecule of glucose yields around 30 to 32 molecules of ATP. This massive boost in energy production is what makes complex life forms like humans possible. Furthermore, understanding this process explains how deadly poisons like cyanide work, as they kill precisely by halting this vital proton-pumping machinery. Let us bring all these concepts together and look at exactly what is happening inside the cell. In this diagram, the horizontal structure is the inner mitochondrial membrane, acting as our biological dam. Below it is the mitochondrial matrix, and above it is the intermembrane space. Notice the protein complexes, labeled 1, 3, and 4. They are actively pumping the red protons upward, creating that high concentration of positive charge in the intermembrane space. Because the membrane is strictly impermeable, those protons cannot just leak back down. Their only escape route is through the large structure on the right, the ATP synthase. As protons flow back down through this specific channel, the enzyme harnesses their return flow to chemically bond ADP and phosphate together, forging the ATP that powers your body. Now that we understand the biological machinery, let us look at the mathematics behind it. The energy stored in this proton gradient can be precisely measured. We call this stored energy the proton motive force, or PMF. The formula for the proton motive force beautifully links chemistry to electricity. Let us break down every symbol. Delta P represents the total proton motive force, measured in volts. Delta psi represents the electrical membrane potential, which is the voltage difference across the membrane. The term with R, T, and F contains the gas constant, temperature, and Faraday constant, which converts the chemical gradient into an electrical equivalent. Finally, delta pH is the difference in acidity across the membrane. In plain English, this formula tells us that the total driving force pushing protons back is simply the sum of the electrical voltage pulling them and the chemical concentration pushing them. If either the voltage or the acidity difference increases, the total driving force becomes stronger. Let us apply this formula with a concrete example. We want to calculate the actual proton motive force in a typical human mitochondrion at normal body temperature. We are given an electrical membrane potential of 150 millivolts. The outside pH is 7.2, and the inside pH is 7.9. At body temperature, our constant term evaluates to roughly 60 millivolts. First, we find the difference in pH by subtracting the outside from the inside, giving us 0.7. This means the outside is significantly more acidic. Next, we multiply this 0.7 by our 60 millivolt constant to convert the chemical gradient into an electrical equivalent, which gives us 42 millivolts of chemical driving force. Finally, we add the 150 millivolts of electrical pull to the 42 millivolts of chemical push. Our final answer is 192 millivolts. Physically, this means the combined gradient acts like a tiny 192 millivolt battery across the membrane. Now that we have calculated the voltage of our biological battery, we need to know how much actual chemical energy it can release. This brings us to the thermodynamic equation that links voltage to free energy. The formula is delta G equals negative N times F times delta P. Let us define these terms. Delta G represents the change in Gibbs free energy, measured in joules. The variable N is the number of moles of protons moving across the membrane. F is Faraday's constant, representing the charge of a mole of electrons. And delta P is the proton motive force we just calculated, but it must be in volts. The negative sign in the formula is very important. It indicates that energy is being released by the system, which is exactly what we need to power the production of ATP. When the proton motive force increases, the resulting free energy becomes more negative, meaning even more energy is released to do biological work. Let us see exactly how much energy is released when 1 mole of protons flows back through the ATP synthase. We know our proton motive force is 192 millivolts, which we must convert to 0.192 volts to make our units match. We are looking at 1 mole of protons, so N equals 1. Faraday's constant is 96,485. We substitute these values into our equation. We multiply -1 times 96,485 times 0.192. This gives us negative 18,525 joules per mole. Converting to kilojoules, our final answer is approximately negative 18.5 kilojoules per mole. Physically, this tells us that 18.5 kilojoules of energy are made available every time a mole of protons flows back into the matrix. This is the exact energy harnessed by the ATP synthase turbine. So, what does this 18.5 kilojoules of energy actually mean for the cell? We have to compare the energy released by the protons to the energy required to build ATP. In biological conditions, it takes about 30 to 50 kilojoules of energy to synthesize 1 single mole of ATP. If 1 mole of protons gives us 18.5 kilojoules, simple division tells us that we need the energy from roughly 3 to 4 protons to manufacture a single molecule of ATP. This mathematical result is absolutely beautiful because it perfectly matches the physical, mechanical rotation of the ATP synthase enzyme observed in laboratories. The flow of those 3 to 4 protons provides the exact mechanical torque needed to click the enzyme into its next position and forge a new ATP molecule. We have covered a lot of ground today, so let us reinforce the most important ideas. First, remember that your cells act like microscopic hydroelectric dams. They pump protons across the inner mitochondrial membrane to create a powerful electrochemical gradient. Second, this gradient stores a massive amount of potential energy, which we can calculate as the proton motive force. Finally, as those protons flow back down their gradient, they pass through the ATP synthase enzyme, physically spinning it like a turbine to forge ATP. This elegant system is the fundamental reason why you and almost all complex life on Earth have the energy to exist and thrive. You now have a complete understanding of the chemiosmotic hypothesis, from the biological concepts to the underlying mathematics.